Our journey now arrives at another essential organelle, floating within the cytoplasm. Unlike the organelles we've explored so far, this one specializes in supplying the energy that keeps nearly every cellular process running. Ahead of us lies the mitochondrion, often described as the cell's power station. As we move closer, the smooth outer membrane comes into focus. It forms the first protective boundary of the mitochondrion, while allowing many small molecules to pass through specialized channels. Crossing this membrane brings us one step closer to where energy is produced. We've now entered the narrow region between the two mitochondrial membranes. Although this intermembrane space appears small, it will soon become central to one of the most important energy-producing processes in biology. Just ahead, the landscape changes dramatically as the inner membrane comes into view. Rather than remaining smooth, it folds inward repeatedly, creating an enormous amount of surface area that allows thousands of energy-producing proteins to work simultaneously. We now travel through these deep folds, known as Cristae. Every curve increases the available membrane surface, allowing the mitochondrion to pack in far more molecular machinery than a flat membrane ever could. Beyond the inner membrane lies the mitochondrial matrix, the innermost compartment of the organelle. This space contains enzymes, ribosomes, and even its own circular DNA, reflecting the mitochondrion's unique evolutionary history. Embedded throughout the inner membrane are large protein assemblies arranged with remarkable precision. Together, these complexes form the molecular system responsible for converting the energy stored in food molecules into a usable cellular form. As we move alongside these complexes, electrons begin flowing from one protein to the next. Rather than releasing their energy all at once, they pass through a carefully controlled sequence known as the electron transport chain. Each electron transfer releases a small amount of energy. Instead of being wasted as heat, that energy is immediately captured by the protein complexes and used to perform another essential task. Using the energy carried by these electrons, the complexes actively pump hydrogen ions across the inner membrane. This process steadily moves protons from the matrix into the intermembrane space, storing energy in the form of a concentration difference. Small mobile electron carriers now shuttle between the larger protein complexes, delivering electrons with remarkable speed and precision. Their continuous movement keeps the entire electron transport chain operating efficiently. As more protons accumulate outside the inner membrane, a powerful proton gradient begins to form. This difference in concentration stores potential energy, much like water collecting behind a dam before flowing through a turbine. Standing before us is ATP synthase, one of the most remarkable molecular machines found in living cells. Rather than using electricity or fuel directly, it captures the energy stored within the proton gradient. The accumulated protons now begin flowing back into the matrix through ATP synthase. As they pass through the protein, their movement is converted into mechanical rotation, transforming stored chemical energy into motion. Looking closer, the rotating F0 rotor spins rapidly within the membrane. Every turn transfers mechanical force upward through the central shaft, linking proton movement to the reactions taking place above. At the top of the enzyme, the catalytic head receives ADP and inorganic phosphate. The rotating shaft continually changes the shape of the catalytic sites, preparing them to build the cell's energy currency. Another rotation completes the reaction, joining ADP and phosphate to produce ATP. The newly formed ATP is immediately released, ready to power everything from muscle contraction and active transport to DNA replication and protein synthesis. Across the extensive folds of the inner membrane, thousands of ATP synthase molecules operate at the same time. Together, they generate an enormous supply of ATP, providing the continuous energy that living cells require every second. Freshly produced ATP now leaves the mitochondrion and enters the surrounding cytoplasm. From here, it travels throughout the cell, delivering usable energy wherever work needs to be done, connecting this remarkable organelle with nearly every cellular process we've explored. As our journey leaves the mitochondrion behind, we carry with us a deeper understanding of how cells transform the energy stored in nutrients into the ATP that drives life itself. That same energy will soon fuel the molecular events waiting in the next stage of our exploration.